Welcome back, all of you. Here we'll discuss today in the second session. We'll discuss about the uh, cell cycle phases and its importance in cancer, and describe the programmed cell death and its role in cancer. Various molecular changes occurring in cancer with respect to DNA, the genetic changes, and various genetic mechanisms by which cancer is caused. So the different headings under which we'll finish this session. And basically, come to the cell cycle. We are not going to very deep into cell cycle. We want to see the places where actually cancer is affecting and how the cancer is going in with the cell cycle perspective. We have four phases: G1, S, G2, and M phases. This order you have to remember because sometimes this can be a potential question for you. Gap phase, and middle of the M and G1 phase, we have G0 phase where the cell goes into a resting state. Okay, we are concerned with the stages which are actually potentially involved in the cancer process. So this is the potential places. G1 checkpoint, I have given you three checkpoints: G1, G2, and M checkpoint. The most important checkpoint is G1 checkpoint and also the M checkpoint. G2 is actually a completion of everything. So the G1 and M checkpoints, these are the places where the cell is decided whether to go forward into division or to stop back. Because if a cell is not large enough, or if a cell is having abnormal DNA at DNA checkpoint, because the DNA can have some errors in replication or transcription, and if in this replication the DNA has gone into some mutation and the DNA is not normal, it is not allowed to go forward, and these are called the checkpoints where the cell is stopped or the cell is when going to the automatic cell death. Okay, unless until the cell is normal and the phase is going normally, and then only it is allowed to go forward. Okay, that is importance of checkpoints in a cell cycle. And coming to the programmed cell death, as I told you, if a cell is not properly moving through in the in these cell cycle phases where there is gap 1 synthesis and gap 2 phases, and it is subjected to programmed cell death. And also any old or senescent cell in the body, senescent means the cell is getting old and it's not able to perform its normal physiological functions. These cells are going into a physiological process called programmed cell death or apoptosis by having some distinct biochemical mechanisms. Okay, and this will be discussed in deep in your pathology part, but for now I'll tell you some changes like protein cleavage, protein cross-linking, DNA breakdown, and phagocytic recognition. These cells which are decided for a normal programmed cell death, they are having more amount of protein cleavage, more amount of protein cross-linking, breaking of DNA, and phagocytes will recognize them as abnormal cells to get to phagocytose them. And these are three pathways which are important pathways which are involved in the apoptosis. You at an undergraduate level in the first year, you should know three important things. One is how many pathways are there: extrinsic, intrinsic, and granzyme pathway. Some books doesn't give you granzyme pathway, but it is important. And second thing, what organelle is executing this programmed cell? It is mitochondria. Mitochondria is the key player of programmed cell death. No mitochondria, no programmed cell death. It's just a crushing in the spleen. Like for example, if we take RBC, there is no mitochondria. RBC are not prone to apoptosis. They are crushed away in the spleen by the small passages. They are not. They are not. There is not a programmed cell death. In the mitochondria, the third point, the first point is number of pathways: extrinsic, intrinsic, and granzyme pathway. Second point is what is the organelle involved, it's mitochondria. And the third point, what are the enzymes involved? Caspases. You name a caspase: caspase 8, caspase 9, caspase 10, caspase 3. These are the different caspases which are activated in a programmed cell death pathway. Okay, these are actually what happens is the cell is going to different a progressive and programmed sequence of events like endonuclease activation, chromosomal degradation, protease activation, and degradation of nuclear and cytoskeletal proteins and cytoskeletal reorganization. Finally, what happens? The cell becomes into a blebs, a small blebs will be there, and cell shrinks in size and forms from apoptotic bodies, and finally the cell is killed off. Okay, this is the programmed cell death. It happens to every cell in the body which has passed its lifetime and the function it's not functioning properly, or sometimes the abnormal DNA during the division can also switch on the apoptosis process. And now coming to the carcinogenesis, overview of it, how it is happening. Like what we have discussed, the carcinogens will stimulate a normal cell and they cause a DNA damage. And if it is successfully repaired, it is going to normal cell. If it is not repaired properly, mutations in somatic cells, and these mutations they activate proto-oncogenes. The impaired apoptosis that is not properly done, inactivation of tumor suppressor genes, and finally giving you abnormal gene products or proteins, and which are regulatory proteins. Finally you land up in a malignant tumor. Okay, now we'll discuss in detail about what is actually happening. Carcinogenesis. The whatever the diagram I am showing you, it's from Harper, and it is giving you the list of proteins or factors which are helping in the carcinogenesis, like growth factor receptors, G proteins, signal transduction proteins, transcription factors, tyrosine kinases, cell cycle regulatory proteins, and apoptotic regulators. These factors, any or all of these factors, they promote a carcinogenesis. The process of carcinogenesis is promoted by either all of this or a few of this. Okay. And coming to the mechanism of carcinogenesis, we have been talking much at the point mutation or a change in DNA that should happen to produce a cancer. Like DNA needs to be modified for cancer to occur, because a normal DNA is not cancer DNA in a cell, and that DNA has these modifications to be taken place to produce a carcinogen. And we this DNA is specifically called as an oncogene. That oncogenes we'll discuss in the next part. But these are the basic elements of a carcinogenesis which happens in the DNA: promoter insertion or enhancer insertion, chromosomal translocation, gene amplification, finally a point mutation. These are the different mechanisms by which the DNA gets modified into a cancer DNA or an oncogene. Yes, coming to the promoter insertion. Retroviral reverse RNA, which is reverse transcribed to the host DNA, can be inserted into the DNA to produce a cancer cell. The complementary DNA flanked with LTR, long terminal repeats. These flanked DNA molecule can get integrated into the DNA and cause the carcinogenesis. Example is c-MYC gene, which is associated with B cell tumors and human colorectal cancers. Promoter, I told you two parts here: promoter insertion and enhancer insertion. Those differences I'll tell you in the next slide. Promoter and enhancer, there are two different things which are being joining to the DNA either upstream or downstream to a particular gene which is not in an active state. Okay, so first thing, I'll give you an example of MYC gene, c-MYC gene. The c-MYC gene, this proviral DNA, I told you, any RNA virus or any DNA virus which can be incorporating its genome into the cell, into the genome of the human being, then it becomes a proviral DNA, and that integrates into the gene. And this MYC gene, which is not a carcinogenic, before an integration of a proviral DNA becomes carcinogenic and produces multiple copies of RNA, mRNA by the insertion of proviral DNA. This is promoter insertion. So, go to the next step. It is enhancer insertion. The promoter insertion, as I showed you in the diagram, it is starting at that point of insertion of the, it is upstream, and it is starting at the point of insertion. The transcript is promoted fully, completely. But enhancer is a bit downstream. It is inserted a bit downstream, and that doesn't start at the exact point of insertion. It will have a lag, a distance between that, and that will be promoted in a different sequence. Okay, there is not much difference in promoter and enhancer. Unless the most subtle difference is one is upstream and one is downstream, as Harper says. Okay, promote and enhance differs in region of binding either upstream or downstream to the particular proto-oncogene what is discussed. Okay, and apart from promote and insertion, there is a classical phenomena called chromosomal translocation, known from ages. What we know is BCR gene and AB. The best example is Philadelphia chromosome, which we'll be talking about in case of CML, chronic myeloid leukemia in kids. Okay, a portion of chromosome splits off and joined to a different chromosome. If you see here, chromosome 9 is having a dark gray limbs, and chromosome 22 is having a light gray limbs. And if you see the translocation part, the 9q, the 9q part of chromosome 9 is getting dislocated and going and attaching to the 22 chromosome. And finally, the BCR ABL product gene, the fusion gene, is causing the leukemic stem cell, or that is causing the chronic myeloid leukemia, the classical example of chromosomal translocation. And if there is Philadelphia chromosome in your in the chromosomal analysis, take it for granted the patient is prone for getting CML. So this helps in diagnosis of early diagnosis of CML in kids. Okay, and apart from this, there is other classical example where I want to add a point here. It is in this chromosome transfer. The first example I told you, it's a chimeric BCR-ABL gene. Chimeric means it's a com composed gene or combination gene. Like BCR goes and attaches to ABL, and that is a composite product, giving you a fusion gene. But coming to this case, Burkitt's lymphoma, it is 814 translocation. It is not like a chimeric gene. It is like activation by attachment of different parts. See here, the 8th chromosome part is going and attaching to the 14th chromosome, and a part of 14th chromosome is coming back to the 8th chromosome. These two chromosomes, there is a reciprocal translocation between chromosome 8 and 14, and then the c-MYC gene is activated, and it is causing Burkitt's lymphoma or nasopharyngeal carcinoma. So these are the different patterns of chromosomal translocations where we have the cancer getting affected. So summarizing, the cell cycle regulation is a critical event of carcinogenesis where the G1, G2, and M phases are the carcinogenic, the potentially carcinogenic checkpoints, where any disturbance in these checkpoints is going to cause a carcinogenic potential. Next, apoptosis. Apoptosis is a programmed cell death and is physiological phenomena occurring in age and senescent cells. And also, apoptosis is helping the cells to degrade or destroy the abnormal DNA cells which is getting an abnormal DNA in replication. And genetic modifications. What are the genetic modifications? Promoter insertion, enhancer insertion, chromosomal translocation, and point mutation can lead to cancer cells. Thank you.